So first of all, thank you, governors, for listening to me. For those of you who panicked at the slide, I promise you that we will take a deep dive into science only in the middle third of the talk. Other than that, I am going to give you a general introduction, and then I'm going to put forward a proposal that a group of us is starting to develop. It's very, very embryonic at this point, and this is the first time that I make this proposal at an international meeting. And there are, as I will tell you, there are similar proposals. In fact, some of Rich's inspiration is going to have to be needed to deal with the fake news of the anti-vaccination movement, which has very similar problems to the GMO movement. So, very briefly, what I'm going to tell you about is the challenge that we have. Today, I'm going to talk to you guys about the part of my laboratory that actually works on RNA viruses, and not the cellular RNA part. So we will talk about these challenges of dealing with what we don't know yet, what's coming around the corner. So I'll give you an example, which is the example we work with, but I'll point out similar examples of other viruses. I will then delve, for the RNA aficionados in the audience, we will have a little bit of fun with RNA biology, but there's a translational, if you like to use that word, bent to this, because our work and work of others has actually shown us a way to make vaccines for flaviviruses. And then finally, I'll go very briefly over this very inchoate idea that a group of us are having to supplement ideas that are ongoing right now for what we call a global rapid biodefense response. So the challenge, as I said, is how do you deal with emerging, re-emerging, and most challenging, potentially emerging epidemics? And I think I have one example here that many of you have seen. This is the years of expected life. And if you were sitting here happily in 1910, you would say, life is great. Life expectancy is rising. Women higher than men. Women are in triangles. But then something happened, or several things happened. But the major contributor to this was the pandemic of influenza. Okay, you can see other aberrancies. You can see here aberrant things that change men more than women. You can probably imagine what that was in the late 1930s. But this is a dramatic crisis that humanity faced. And the problem is that we know we're going to face it again. We don't know what it is yet, but we know we're going to face it. Beyond that, this is an old slide from a paper published in Science in 2000 about another great pandemic of the 20th century, the HIV pandemic. And this is one of the reasons that I find this type of disease so depressive and at the same time so interesting to study, is that it actually changes our demographics in ways that affect the young, the productive. I don't know how well this prediction is holding off. This is the population of Botswana as it would have been if AIDS was not there, versus the population as it was predicted to be based on mortality from HIV. Now, this preceded the use of highly active antiretrovirals, so this may have been somewhat changed. Nonetheless, very few things in life change a population pyramid like a pandemic of this sort. And I have to say that, again, when you're thinking of where do we stand now versus where the crisis is going to be, when I was in medical school many years ago, a terrific textbook of virology by none other than Darnell, Luria, Baltimore, Nobel laureates. It was a great textbook of virology, and it said, there has been not a single retrovirus associated with human disease. This was obviously before this started. Nonetheless, it did say there are retroviruses that cause leukemias in cats and other animals. So these two examples that I gave you are both examples of how we are sitting here and we don't know what's coming in a few years. Yet we believe we actually have to be ready for it. We have to be as ready as we can be. And the only way that I know how to be ready for it is through science and understanding these diseases. I am going to focus on RNA viruses for many reasons. The two examples that I gave you, the influenza pandemic, I don't like to call it the Spanish flu because I'm half Spanish, but the influenza pandemic of the early 1900s and HIV are both RNA viruses. And I think we can make a long argument that RNA viruses have an uncanny capacity to cause problems because they mutate very, very quickly, because essentially even when you're infected with one virus of hepatitis C, by the end of your infection, you probably have more different viruses in your body, different genomes, than 10,000 people's infections with other viruses. So these viruses are particularly problematic, and history tells us, as we are seeing today in Africa with the ongoing Ebola outbreak, that these RNA viruses are a real problem. So I'm going to take the example of flaviviruses to discuss some of the ideas that we have and tell you about 3 flaviviruses that we work with and about how our studies and studies of others on a particular viral RNA have taken us to propose a general principle to make vaccines for these. And then finally, the initiative I told you I would bring forth. So the giant among these flaviviruses that affect humans is dengue. Not because it's a particularly lethal virus. I had it as a kid, I survived it, I wasn't even hospitalized. My sister, on the other hand, was in the hospital. It's because of the numbers. Dengue probably, and this is only an estimate, because the majority of people who actually get infected with dengue don't have any symptoms. Yet it can be very severe, and it creates an enormous loss of money, time, and it creates a lot of suffering. There's probably 400 million infections a year. And Rich mentioned some of these mosquitoes. Some of the ones that I wouldn't mind being eradicated are some of the Aedes family mosquitoes. Aedes aegypti, which transmits the 3 viruses that I'm going to tell you about today. And personally, I'm not sure the world would be a worse place if there's no more Aedes aegypti around. First of all, they're like dogs. They're domesticated. They actually live with us now. They've learned to live in our houses, in our potted plants, in our plastic. So getting rid of plastic may actually help another thing. So today there is an approved tetravalent vaccine. And I don't want to get into it, but it's extremely, extremely limited in where it should be used and in its efficacy. There are no approved specific antivirals for dengue. So then everybody remembers, just a few years ago, even though we have a short memory, there was a Zika pandemic, which really should not have caught us by surprise. This is one of those examples of you're here and you don't know what's going to happen next year. We should have seen it coming, but we didn't. It spread like wildfire, especially in the Americas, and it actually presented us with a series of problems that we hadn't experienced with these types of viruses before. The most important is the genitourinary tropism of this virus. That is involved both in its sexual transmission and its vertical transmission from the mother to the fetus. Tremendous amount of focus has been made on the neurotropism, but that's really particularly only during pregnancy. So, and then the third virus, which happens to be intellectually my favorite, although it's the most lethal, is yellow fever virus. Now, yellow fever is an old enemy, but it represents a huge global threat. Why? Because it has very, very high mortality. It can be as high as 30% among those of you who are of Caucasian origin. And it is, I think, a very interesting example of lack of will, of social will. There is a superb vaccine that was developed in the 1930s at Rockefeller University by Max Tyler. That vaccine is probably one of the, if not the best vaccine in the world, and yet 30,000 people die every year because of yellow fever. There are not enough doses around the world. You all know when there was an outbreak in Africa recently that they have to start testing, giving fractions of a vaccine for yellow fever. This is mostly for the Americans in the audience, very few of us here, but it talks about two American Yankees, I think they were both called Thomas, who went to the south of the United States, and most people in the US have forgotten that yellow fever was a huge problem in the United States. Both died. One died in North Carolina, one died in Texas. This virus is very dangerous because the majority of the people in the world who are very near where it is are not vaccinated. A nightmare scenario for me is a transmission of yellow fever into the Caribbean, where it hasn't really been seen, or presumably hasn't been seen in decades, where people would not even, doctors wouldn't recognize it. A doctor in San Juan, Puerto Rico would not know what a yellow fever case looks versus a severe dengue case until somebody becomes jaundiced. And then from there to Miami. But even bigger worry for me is what if it goes into India? Yellow fever has never really taken hold in Asia for reasons that we don't completely understand. There were importations from Africa recently, but it is a huge concern. It's transmitted by the same mosquito that transmits dengue and that transmits Zika, primarily, which is Aedes aegypti. So this is a global threat. I don't know what the population in India that's vaccinated for yellow fever, but I would venture to guess that it's less than 0.01% of the population. In San Juan, it's probably less than 0.05%. So this is a problem. We are not ready for this, although we could be. So we also study these viruses because they're RNA machines. They're RNA protein machines, and I am fascinated with RNA protein machines. And I'm not going to belabor all of my love for RNA structures, et cetera, but to tell you, these viruses are really, really simple. Their genome is a single-stranded RNA, which is positive sense, so it's also a messenger RNA. It encodes for one of the most complex proteins that you could imagine. This protein actually challenges the translational machinery of the cell. And here I mean translation in the real sense of the word translation. And there's a whole project in the lab trying to understand how does the ribosome in our cells is capable of dealing with making this enormous protein that has 22 transmembrane domains. But today we're going to focus more on the RNA side of the world. It has, the RNA has a 5' UTR, a 3' UTR, a cap. It doesn't have a poly A tail at the end. So it looks like a very specific type of messenger RNA in our cells, which are the histone messenger RNAs. They look very much like this. They don't have a poly A tail. This RNA comes in contact with many different parts of the cell. There are some people who claim that it may even come into the nucleus in some types of infections. I'm not a believer of that, but in this cartoon of the viral life cycle, you can see that the RNA is interacting with membranes. It grows in these packets where it replicates. It interacts with the machinery to be packaged, to be thrown out again into the outside of the cell for the next infection. But today we're going to be focusing on one very specific thing that this RNA genome does. And that very specific thing is that the majority of the genomes in every infected cell, when you get dengue or you get Zika, this is what happens when you walk around, or you get yellow fever. There's always, it was the plant. That GMO, it was GMO, I bet you. Exterminate the thing. What happens to the viral genome is the majority of these genomes actually get processed to form a non-coding RNA. And you know, it's now even in the textbooks that there is a revolution in biology, that there's a lot of biology being regulated by non-coding RNA, RNAs that are not messenger RNAs, RNAs that have nothing to do with making a protein. I think Mauro will tell you some really exciting stories of small versions of these things. So we're gonna focus on this non-coding RNA for a few minutes. And I have to give credit to the people who did the work, because it's been a while since I've actually put a pipette in my hand. So Alex Ward, an American postdoc, Catel Bidet, a French graduate student, Julien Pompon, originally a fellow in the lab, now my co-PI in Singapore, Ruben Soto Acosta, Mexican postdoc, who's now in Minnesota, and Shisha Ye, who is a postdoc who's now in Taiwan. So almost as international as the ICGEB labs. Not quite, but almost. So this guy started being very interested in this RNA. So this RNA is formed by the most inefficient of mechanisms. You get an exonuclease that essentially chews down the whole 11 kilobases of this virus, but then meets these really beautiful structures in what is this 3' UTR and stalls. And it can stall 4 times, about 4 times. And this is a very nice review by Jeff Keefe from Colorado that summarized it in a very nice cartoon way. Here you see the exonuclease is dead, can't go any further, so it's stalled in there. And you can actually purify a complex with the exonuclease at the end. And as I told you, this non-coding RNA accounts for a large portion of the RNA in an infected cell. So the virus sacrifices sometimes 90% of its genome to make this small RNA. Well, medium-sized RNA. I hate the terminology, by the way. So this is what the cartoon looks like of this RNA. It actually has these 2 very similar structures here, and then 2 others that look very similar. These are called dumbbells. These are called RNase-resistant regions. And intriguingly, this whole region comes from what was called traditionally in the flaviviral field, the variable region. If you sequenced all the flaviviruses in the world, whether they were only mosquito or insect or plant or human, this was the most variable region. Yet all of them, and now this has been very nicely documented by a student that I mentored in Singapore, who's now in Switzerland, it has been documented phylogenetically that they all form very, very similar structures, but with very different sequence. So we became fascinated with this RNA, and Catel and Alex did a really, really important experiment. This experiment has been the foundation for almost everything that I'm going to tell you about. And this is almost as technical as I'm going to get today, okay? So you all know that interferons are part of what we call the innate immune system. It's a very powerful antiviral system. Without interferons, we would be dead. In fact, recombinant GMO interferons are actually made and used. So this was an experiment. If you got rid of the proteins that were most likely to bind this RNA, you crippled the immune system. So these 3 proteins were found to consistently, every time you had infection of dengue 2, these 3 proteins, which I'm not going to bother you with exactly what they are, suffice it to say that we discovered they're part of the innate immune system. And what they do is they change the way the cells respond to interferon. If the proteins are not there, you essentially have almost no response to interferon. So the conjecture, after many other experiments, that Catel and Alex came to was that the role of this small RNA was equivalent to knocking down these proteins. This small RNA inhibited these proteins, it gave the virus the advantage, and then the virus could replicate. If the virus has a deficient non-coding RNA or cannot make much of it, then these proteins are made. These proteins are part of the innate immune response. The innate immune response wins, and the infection is not effective. So fast forward a couple of years and a great collaboration with my colleague, and I'm going to be careful with the plant, En Young Oi. In a paper we published in Science a few years ago, we actually showed that variations in this non-coding RNA could explain the epidemic behavior of dengue. So just essentially 3-point mutants, and at the end of the day, it's only these two right here. These variants in the virus could explain the difference between a virus that was endemic in the Caribbean versus one that became epidemic and was actually more severe. And we could understand the mechanism. It again had to do with another way of controlling interferon production. And so this tied for the first time this non-coding RNA to epidemiology, to epidemic potential of the virus. So does the same thing happen with Zika? And I'm not going to belabor all the data, but I'm going to tell you that the answer is yes. And in this case, I'll give you another example. It's a different protein called FMR. FMR is a protein that binds this RNA, this non-coding RNA, and the non-coding RNA inhibits the protein. We've shown this both in human cells and in animal models. But what I want you to focus on is this particular mutant here, created by my colleague Pei-Young Shi and published in Nature Medicine. This mutant is actually the leading attenuated vaccine for Zika right now. It's the one that has performed the best in non-human primates, is in clinical trials right now. The NIH is taking it in clinical trials. And what you can see by looking at this northern, and yes, we still do northerns in the lab. For those of you who we have gone back. You can see that one of the things that's very noticeable about this mutant is that it makes very, very low levels of this non-coding RNA. Okay, so this is the general model for how this is working with Zika. We actually know that it's repressing FMR, and that FMR in turn represses translation of Zika. So when you don't have the sfRNA, FMR wins. When you have the non-coding RNA, the non-coding RNA represses that and permits stronger translation. This was just published in eLife last November. But the beauty of this work, in conjunction with the beauty of Pei-Young Shi's work, and going back to the literature, to an old vaccine developed by, a dengue vaccine developed by the NIH, which inspired Pei-Young, is to suggest that any time that you cripple that non-coding RNA, you have a highly attenuated virus. One that still can replicate. The mechanics of the virus are fine, but it cannot deal with the innate immune system. So we believe, and I'll bring that up again later, that right now, if we discovered any new flavivirus in a matter of a week using new recombinant technology, we would have a vaccine candidate ready to be tested in non-human primates, if necessary, which is remarkable. And I give Pei-Young Shi tremendous amount of credit because he's the one who's deployed the technology to do it that quickly. So our model, back to just the science, is that this variable RNA is a sampler of human proteins that are involved in innate immunity and innate restriction. And that essentially the virus evolves these parts of the virus that it doesn't need for its mechanics. So it doesn't need that part of the 3' UTR to do anything else. It translates equally well, it replicates equally well. So it is free to evolve variation. And why does it evolve? Because it changes the host populations that it meets. So this variable region then in many different viruses, and Dengue 2, it uses these proteins. In Zika, it likes FMR. This variant of Dengue 2 now attacked TRIM25, another interferon producer. And by doing that, it actually reduces innate immunity. And that's how this virus evolves to deal with our innate immune system. So an immediately obvious question was, what about mosquitoes? And this is a slide that I usually show to show. This is the slide that I show, but it's embarrassing enough, so I've decided to use this slide today. No implications here with this Let's see if it stays still. No implications about any kind of connections here, Rich. But we now know flavies can actually go from one person to the other, which is something that we didn't know in humans before. But really, the other host of this virus is Aedes aegypti, of these viruses. Sometimes they can be transmitted by Aedes albopictus, but really Aedes aegypti is the monster vector. So half of the lab works on Aedes aegypti. So the answer to the question is, what happens to this non-coding RNA? So other people, Andrea Gamarnik from Argentina, a tremendous scientist, and others, showed that the sfRNA is made in mosquitoes. And in a paper we published in PLOS Pathogens in 2017, Julian and the lab and I showed that just like in humans, the RNA in mosquitoes is there to inhibit the innate immune system, or at least that's one of its big functions. And actually we could show that different viruses with mutations in this RNA would actually deal better or worse with different mosquito backgrounds depending on the toll pathway of mosquitoes. So that was very cool. But when you're doing a study, there's always a tremendous surprise. And the biggest surprise of us was that when we actually, and when I say we, I mean Shisha, who was an expert, dissected these mosquitoes, she noticed something very, very interesting. She noticed that the salivary glands had by far the highest level of sfRNA, of this non-coding RNA. The highest level, the ratio of the sfRNA to the genomic RNA, which was obviously very interesting because the salivary gland is the last organ before transmission. It produces the virus by expectorating the virus into the saliva, which then gets injected by accident while the mosquito's probing around the capillaries. So we became very fascinated by this idea, and I'm just going to show you one piece of data on this, but first I'll give you the model. So we thought, okay, why is this so high in salivary glands? Well, it makes sense if you have a high level of salivary glands. We already know that actually, I didn't tell you, but we know it inhibits the innate immune system in salivary glands, not only in the whole mosquito, but in salivary glands. And that would then say you can replicate the virus more. That's great. And then you get more virus in the saliva. And in fact, we do see a small increase of virus in the saliva in those mosquitoes that are more epidemically fit. So we were able to go back to the Caribbean epidemics and take different isolates and use if you wish, the population genetics of the virus to show that. But the more intriguing idea, and I wouldn't tell you if I didn't think I was right, but we're not 100% yet sure, is that maybe the sfRNA actually itself goes into the saliva, and that it's help in transmission, that it actually helps by affecting the cells at the biting site. This is not new. I thought this was so new. You start looking in the literature, all kinds of parasites, battling hosts, have used small RNAs to attack the host and vice versa. So the bottom line is that we have now very nice evidence that this may be true. So first of all, we can detect sfRNA in the saliva itself. We can detect it by RT-PCR, but we can also detect it by sequencing. And we know that 2 of those 4 versions of sfRNA we can find in the saliva. They're the 2 most prevalent versions. So it could be in one of 2 forms. It could either be inside some vesicle. We don't think it's inside the virus, and we have some data to show pretty carefully that it's not inside the virus. It's never been found inside variants. So it could be in an extracellular vesicle. Or more recently, there was a beautiful paper about this B protein that actually binds RNAs and keeps RNA stable, and the bee can transmit the RNA into flowers. So we don't know exactly. We believe it's the former for the following reason. We've identified the CD63 homolog in mosquitoes. CD63 in mosquito cells and in mosquitoes is absolutely essential for extracellular vesicle or exosome production. So when we can knock it down in the mosquito, so this is an RNAi experiment where we can knock down CD63. And in the salivary gland, we see absolutely no effect. These are populations of mosquitoes. These are not isogenic organisms. These are really inbred but diverse populations, so you get a lot of variation in your infections. However, there's a dramatic effect in the sfRNA to gRNA ratio in the actually expectorated saliva, suggesting strongly that this sfRNA is moving into the saliva in extracellular vesicles. We already have data that actually if you use that to infect, if you use that sfRNA to treat keratinocytes, which are the primary cell infected, that you actually enhance infectivity. And right now, the killer experiment is ongoing, which is a series of non-human primates that are being infected with either viruses made from these mosquitoes or viruses made from those mosquitoes, where we have the same amount of virus, but in one case we don't have the sfRNA or much lower levels of the sfRNA in the saliva. And this was a real tour de force by Shisha, because you can imagine that every one of these data points is either 5 salivary glands or 5 expectorated salivas, 10 nanoliters each. They go into a 10 microliter tube. So it is an amazing amount of work, but very important. So we believe that this mechanism is actually important both for human cells as it is for insect cells. So this is basic discovery science, right? But we think it actually, as I mentioned before, has important implications. So this work and very beautiful work of others like Andrea, like my colleague Ian Young, like Mike Diamond and others in the United States have inspired us to think that we really need to use this basic knowledge that we're getting about viruses to actually do something which we call more like actionable epidemiology. I really don't want just to know that the virus will come to Colombia next week or that it will move to Panama. I want to be able to do something about it. And I'm not alone. Obviously, there are many, many people. Many of the initiatives out there are really focused on one virus or one family of viruses. For example, Nipah Hendra. But the problem is that we really don't know what the next pandemic is. So what we're proposing is, first of all, would require immense international collaboration. And I can tell you there are some great barriers to this, because there's distrust between politicians. So I think this is one place where I like what Rich said. There should be more science into making these decisions and less politics. So the bottom line is that up to now it's a very small group, but we're starting to meet with stakeholders around the world to try to create essentially what would be a repository of knowledge, and then to create new knowledge in the following areas. So we need evidence-based epidemiology, and that has to consider both the host and the pathogen. And I'll give you an example immediately. We need to start creating generic vaccines. These generic vaccines are not going to be the best vaccine you're going to make, right? They're going to be the vaccine that's going to diminish the disease enough to get things stopped. And that's, you know, this adage, which I never thought I was going to invoke, but the perfection is the enemy of excellence. Is what we need many, many times is a vaccine that will diminish things sufficiently so that then you have time to work. We had no time with Zika. Even as much as we knew about flaviviruses, it was really remarkably fast. So the same thing can be applied to therapeutics, both small molecules and biologics. And the same thing, and I'm glad Rich mentioned this, because I have a slide on this genetically modified mosquitoes, which are already being released if you consider Wolbachia infection and modification, and other modifications that are being released in many countries. And then we need to very, very seriously cooperate with veterinarians, because a lot of these diseases are going to be coming from animals. So this is the first point. Epidemiology needs to consider the differences in the host. Right now, with the UK initiative, with the 1,000 genome projects, we have an enormous amount of information about differences in our genomes. And for example, there is no place where there's larger genetic variety than there is in Africa. And we have tools to actually understand this, both from the point of view of clinical studies, but also techniques such as what's called in vitro GWAS, where you make immortalized cells from individuals and use those to tell you about differences between individuals. This is an example in the US. This was a yellow fever, massive yellow fever epidemic in the US. A third of the city died, a third left. This epidemic changed the history of Memphis and made Atlanta the predominant city in that central part of the southern United States. This is the case fatality rate for people of African descent. This is the case fatality rate of people of European descent. This has been reproduced. We have a lot of historical and more modern data. And on the average, the case fatality rate for Caucasians is 7-fold higher. There are many diseases where it's the other way around. There are many diseases that vary, but understanding the genetic vulnerabilities of the host is critically important. And very soon we'll be able to do that very accurately. As I'm not going to belabor this, the ability to generate generic vaccines based on specific molecular vulnerabilities. We know now this for flavies. Any new flavy, we can try this. We don't know it yet for coronaviruses. We don't know it yet for alpha, for chikungunya, but we're working on it. So that would be, I think, really important. There are other groups much better than we are in the alpha virus world that probably have what we need to know already. So the other thing is, and this is an example of our work too, immediate, rapid screening of already approved or already advanced therapeutics that are already in clinical trials that are known to be safe. Zika was a particularly big problem because of pregnancy. But you could have treated men so they don't transmit it to their spouses, because we knew very early on that it could be transmitted. So any man of a pregnant woman, we actually came up with a bunch of very safe drugs that if I would have been married to a woman who was pregnant and I thought I had Zika, I would have taken that medication. So that is something we can do. Are they going to be the very best drugs ever? No, but they're going to be useful. So the other thing is really a genetic understanding of the pathogen. We and others, now many, have done these massive screens for what are the host factors that these viruses need. We know for flaviviruses now, we have a very good idea of what pathways in our cells they need from work of our own lab, Errol Figuerig at Yale, you name it, many other individuals. And not only do we know that for flavies, but very recently, we just published, this just came out this week, we also have done it for the Paramyxoviridae and the Pneumoviridae. These are very important human pathogens, including human respiratory syncytial virus, measles. You have seen what's happening because of the fake news of the anti-vax campaign. And these are serious diseases. Moreover, these viruses that we studied are all part of a very large family called the mononegavirales. And right very close to human respiratory syncytial virus is Ebola. So even though nobody has done a screen yet for Ebola, we have enough inference from having studied viruses in all of these families to predict what are going to be likely targets for therapy. Again, these are not the optimal drugs, but they're a start. That should be tested immediately. And finally, I'm not going to belabor this, we and others are working on modifying mosquitoes, trying to get them either totally dead or dead when they get infected. This is a particular idea we have of dead when they get infected. So for those who are worried about Aegypti disappearing, I'm sure I have the same reaction to them, and I wouldn't care, but I understand some people may. This will only kill them if they get infected. And then finally, we really need to understand and maybe apply some of these techniques to animals in the wild and animals that surround us. We have to get all these groups together. And what I have to say is that this center, ICGEB, can play an amazingly important role. If you think about Mauro's vision of what we are now, and as Lawrence takes it forward, many of these things fall smack into what this center can do. So this center can be one of the conduits by which this initiative can go forward. I want to thank the people who did the work in my two labs, the lab in Galveston and the lab in Singapore, funding agencies and some unbelievably great collaborators, and you guys for your attention.